This is lecture 9 of 10 in this course on antibiotics, and the topic is antibiotic resistance. The primary learning objectives are first, to understand the major mechanisms of antibiotic resistance. Second, to be able to interpret antibiotic susceptibility reports. And last, to identify and prevent physician and patient behaviors which contribute to antibiotic resistance. To understand the mechanisms of antibiotic resistance, let's first take a look at the major steps that must occur for an antibiotic to act against the bacteria. So here is a bacterial cell with its nuclear material loose within the cytoplasm. Here is some target for the antibiotic. It could represent a ribosome or an enzyme involved in DNA replication or in cell wall synthesis, or any one of a dozen other critical processes. And here are some antibiotic molecules. For the first step, the antibiotic must enter the cell. Antibiotics can make use of bacterial proteins called porins, which span the cellular membrane and allow the antibiotic molecules to pass through. In the next step, the antibiotic must accumulate to a minimum concentration within the cell. Once that minimum concentration is achieved, the antibiotic begins to act against its target in sufficient numbers to cause adverse effect on the cell, and it either kills the cell directly or alternatively prevents its further growth and allows time for the body's immune system to kill it. In antibiotic resistance, the bacteria's genome carries either an altered version of a prior gene or a brand new gene which disrupts one of these three steps. The first possible change is decreased membrane permeability to the antibiotic. This can occur either as a result of decreased expression of porins, a physical change in the porin proteins that prevents translocation of antibiotic, or some other alteration in the physical properties of the membrane. The major antibiotics affected by decreased permeability are the beta-lactams and aminoglycosides. A different alteration with similar effect is the production of another transmembrane protein which acts as an efflux pump to actively move antibiotic back out of the cell, thus keeping the intracellular concentration of antibiotic relatively low. This is commonly seen with resistance to tetracyclines, quinolones, and macrolides. Some efflux systems are specific for a single class of antibiotic, while others work on multiple different classes. The next potential mechanism is an altered target. Whichever protein or enzyme was originally the target of the antibiotic is changed in some way which either makes binding of the antibiotic physically impossible or at least kinetically unfavorable. Changes can include either a slightly different amino acid sequence or some other alteration to the native sequence, such as methylation of a particular amino acid. This mechanism of resistance can be seen with almost every antibiotic class. Finally, the resistant bacteria can produce a new enzyme that inactivates the antibiotic through either cleavage and subsequent drug destruction, or a simple modification which prevents it from binding to its target. The primary mechanism of resistance to beta-lactam antibiotics is enzymatic inactivation by a diverse group of enzymes collectively called beta-lactamases. It is suspected that they originally evolved to combat naturally occurring penicillins prior to the discovery of antibiotics. Supporting this theory, they were first described in 1940, before penicillin had even entered into widespread clinical use. To remind you about the beta-lactam superclass of antibiotics, here are the basic structures of the four types: penicillins, cephalosporins, carbapenems, and monobactams. You will see that they all contain the same four-member nitrogen-containing ring in the middle. All beta-lactamases share the ability to cleave this core ring at the identical spot, which renders the antibiotic completely inactive. Beta-lactamases are as varied as the beta-lactams themselves, with multiple classification systems and nomenclature. Some are called cephalosporinases if they are active against the cephalosporins. There are also carbapenemases, active against carbapenems. These frustrating enzymes are continuing to co-evolve, so to speak, with our increased armamentarium of beta-lactam antibiotics. So how do bacteria gain this antibiotic resistance? There are two main possibilities. The first is called vertical transmission. This is when a spontaneous chromosomal mutation occurs that confers resistance, initially affecting a single bacterial cell, which is then able to multiply as its competition is inhibited or killed by the antibiotic. This generally can result only in the altered target or altered permeability mechanisms of resistance, as an efflux pump or inactivating enzyme requires the addition of an entirely new gene. The second possibility is predictably called horizontal transmission. This occurs when a plasmid or something called a transposon that contains one or more genes conferring antibiotic resistance is transferred between bacterial cells and occasionally even between bacterial species. At this point, I will move on to the next topic, antibiotic susceptibility testing. Susceptibility testing is done to determine the presence or absence of acquired resistance to a variety of antibiotics which might be considered in the treatment of a particular infection. Information the testing will provide for each antibiotic tested will include, for all testing methods, a qualitative result for the isolate in question. That is, the report will indicate whether the isolated bacterial strain is susceptible, resistant, or intermediate to a particular antibiotic. And with quantitative methods only, the report will also indicate something called the MIC for each antibiotic. MIC stands for minimum inhibitory concentration. Standardization of susceptibility testing is governed by a nonprofit organization called the Clinical Laboratory Standards Institute, or CLSI. The CLSI publishes guidelines and reference manuals that guide not only laboratory technique, but also provides recommendations regarding which antibiotics should be tested for which bacterial isolates, as well as what degree of growth inhibition or what MIC should be considered susceptible versus resistant versus intermediate for a particular combination of bacterial species and antibiotic. These guidelines are updated annually, and they incorporate information that comes from in vitro microbiologic studies, pharmacokinetic data, and large clinical trials. The MIC can be a confusing topic when discussing antibiotics. The specific value of the MIC is generally only important for life-threatening and or difficult to treat infections, such as endocarditis or meningitis. While as a general rule, the lower the MIC is, the more susceptible the bacteria is to a particular antibiotic, there is no one cutoff that is universally applicable. The MIC that separates susceptible from resistant bacterial strains varies depending upon the specific organism-antibiotic combination, the site of infection, and the type of media used during testing. As a consequence, accurate interpretation and utilization of the MIC while treating a severe infection usually requires consultation with a reference guide, the microbiology lab, or an infectious disease specialist. As a general rule, a bacterial strain that is described by the susceptibility report to be intermediate to a particular antibiotic should be considered resistant unless there are no other alternatives, the site of infection is a location where the antibiotic concentrates, such as in the urine, or a higher than typical dose of antibiotic can be safely given in a particular patient. There are many types of antibiotic susceptibility testing. I'll talk about these 4 during this lecture. Both broth dilution methods and automated systems are quantitative methods, which can provide MICs, while disk diffusion is qualitative and can generally not provide reliable MICs. To provide a conceptual understanding of how susceptibility testing works, I'll briefly review the concepts behind the first 3 listed here. Although the general category of automated systems are overall the most widely used method in the US, the specifics of how these systems work vary significantly between machine models and have a complexity that is well beyond the scope of this course. As a general category, automated systems are significantly faster and require less labor than manual methods, but are only able to test a limited range of organisms, specifically those which grow easily and quickly. Other organisms require one of the older, more traditional methods. The oldest of these traditional methods is broth macrodilution. The lab technician starts with a nutrient-rich solution of a known concentration of an antibiotic, typically 32 micrograms per milliliter. The technician then makes a 1 to 2 dilution, resulting in a second solution of 16 micrograms per milliliter. The process is then repeated several more times. A known quantity of suspended bacteria are then added to each solution. The tubes are then incubated for about 24 hours. The following day, what will be seen is that some of the tubes will have become cloudy due to bacterial growth, and some will remain translucent. The lowest concentration of antibiotic that inhibited the bacterial growth is then considered to be the MIC. Although broth macrodilution is conceptually very simple, it is very labor intensive and no longer considered practical in high-volume microlabs. A more recent alternative is microdilution. There are different forms of microdilution with various specific parameters varying between manufacturers. However, all use the same basic principle and setup. One starts with a small tray, roughly the size of your palm. There are 96 tiny wells in the tray, arranged into 8 rows and 12 columns. Although this is not consistent, in this particular case, 8 different antibiotics will be arranged in rows A through H, and they will have varied concentrations in the solution in each well, ranging from low to high as one moves through the columns left to right. You can see that the wells all start out as one color. A tiny fixed quantity of suspended bacteria is added by machine to each well. The tray is then incubated overnight. The presence of any bacterial growth then triggers an observable color change. The MIC is considered to be the lowest concentration of a particular antibiotic for which no color change is observable. In comparing antibiotics, in this particular example, we can see that the bacterial isolate is very sensitive to antibiotic C. That is, even when antibiotic C was at a very low concentration, bacterial growth was still inhibited. Conversely, the isolate is highly resistant to antibiotic E, as it failed to inhibit growth even at maximum concentration. The major advantage of the microdilution method is that it is much less labor intensive than macrodilution. The disadvantage is that there is much less flexibility in what antibiotics are tested for a given isolate, as the trays are typically purchased pre-filled. The last traditional method I'll discuss is the disc diffusion method, also known as Kirby-Bauer testing, after the two University of Washington microbiologists who first described it in the 1960s. In this method, the technician starts with a petri dish of agar, which is a generic solid nutrient-rich growth media for bacteria. A known quantity of bacteria is added to the top of the petri dish and spread evenly across the surface. Then commercially available discs or wafers, each of which contains a specific antibiotic, are placed on top. As the antibiotic diffuses out into the surrounding agar, it will inhibit growth of the bacteria surrounding it, if they are sensitive. The agar plates are then incubated for a day, at which time they are re-examined. As you can see, growth will be inhibited to varying degrees around some discs, but not others. The width of clearance around each disc is measured to the nearest millimeter, and based on published tables incorporating diffusion rates for each antibiotic through the agar, that measurement is translated into either susceptible, resistant, or intermediate. The major advantages of the disc diffusion method are its simplicity, low cost, and high flexibility in the choice of which antibiotics to test. The major disadvantage is that it is relatively qualitative. Although some tables exist which attempt to translate clearance zone sizes into specific MICs, this is generally felt to be less reliable than macro and micro dilution. Another disadvantage is that antibiotics with a very high molecular weight, such as vancomycin and the macrolides, diffuse through the agar very slowly. This results in a quantitatively tiny difference in the sizes of the inhibition zones, discriminating susceptible from resistant strains, leading to possible interpretation errors. Now, once antibiotic susceptibility testing has been completed by the microlab, How is that information conveyed to clinicians? It's generally provided in an antibiotic susceptibility report. I'm afraid I don't know the exact format these take in other parts of the world, but in the US, they typically look something like this. How should one interpret this? First, the date and time listed, unless otherwise specified, is the date and time of the original sample collection and not the date and time of the report itself. The source is obvious. There will often next be two separate lines regarding the species identification. The first line indicates the first information that was released from the lab, with the second line indicating a refinement in the initial information. For example, in this case, if the urine was collected on April 4th, the lab would likely have known about the presence of gram-negative rods on April 5th and would have released that information to the clinicians at that time. By April 6th, enough additional testing would have been done to further identify those gram-negative rods as Pseudomonas. The 100,000 CFU, or colony-forming units, is a rough marker of the bacterial load for a particular sample. The lower the number, the more likely one is dealing with a contaminant or colonizer, while the higher the number, the more likely one is dealing with a pathologic infection. So far, the information is relatively straightforward. It's the next section that can be confusing. There is a list of anywhere between 4 and 12 antibiotics, sometimes listed alphabetically, sometimes by class, and sometimes it seems random. Often the listed antibiotics are only a subset of the antibiotics that were actually tested. Many hospitals have protocols in place by which the broadest and/or newest antibiotics are not initially reported here, in order to steer clinicians away from using them if there are older or more narrow options that are acceptable. Next to each antibiotic is a number which represents the MIC in micrograms per milliliter. Although automated susceptibility testing systems don't necessarily need to report MICs in factors of 2, they typically do for standardization across labs and to aid in comparison between individual clinical data and the primary literature. Next to each MIC will be the interpretation of that MIC with an S for sensitive, R for resistant, or I for intermediate. Let's look at what conclusions we could draw from this report. First, the patient has MICs listed for Cipro and Levo that are greater than or equal to some number, which is then interpreted as resistant. So obviously the Pseudomonas is quinolone resistant. The reason for the greater than or equal sign is because the lab, or machine if using an automated system, realizes that there is no reason to identify the exact MIC once it's been established to fall into the resistant category. We see here that there is an I next to gentamicin. As I said before, a report of intermediate sensitivity to an antibiotic means that antibiotic should generally be avoided if possible. This leaves amikacin, cefepime, ceftazidime, imipenem, piperacillin tazobactam, and tobramycin all as possible antibiotic choices. The exact choice will depend upon the patient's comorbidities, such as renal dysfunction, on preferred dosing frequency, any known drug allergies, the general side effect profile of each drug, and the hospital's list of formulary medications. In this particular example, if the patient's renal function was fine and he or she had no allergies, I would consider either ceftazidime or piperacillin tazobactam as the agents with the best balance between not being excessively broad while not exposing the patient to the risk of nephrotoxicity seen with the aminoglycosides. There is one more interesting point to make about this report. You may have already noticed that the MICs for ceftazidime and ciprofloxacin are overlapping, yet the interpretation of the MIC for each is different. Remember that equal MICs between different antibiotics does not mean equal susceptibility. There is one more very specific antibiotic susceptibility issue that I need to discuss. That is inducible clindamycin resistance in Staph aureus, specifically in MRSA. In contrast to healthcare-associated MRSA, community-acquired MRSA is frequently sensitive to various oral agents, including trimethoprim sulfamethoxazole, clindamycin, and doxycycline. Clinical failure of clindamycin is commonly believed due to inducible resistance, which is not identifiable by conventional susceptibility tests. Inducible resistance occurs when the presence of a macrolide triggers expression of an enzyme encoded by the ERM gene that alters the antibiotic target in the 50S ribosome through methylation. This results in a collective resistance to macrolides, clindamycin, and group B streptogrammins, as they all share the same binding site. This is known as MLSB resistance. There are three different phenotypes related to MLSB resistance. The first is the null phenotype, meaning there is no resistance at all. The second is the constitutive phenotype, meaning resistance is always present. The third is the inducible phenotype, in which resistance is only present if triggered by something. The ERM gene is absent in the first of these phenotypes and present in the second and third. In the null phenotype, the Staph aureus bacteria is clindamycin sensitive. In the constitutive phenotype, the bacteria is clindamycin resistant. With the inducible phenotype, it was initially thought to require exposure to a macrolide, which triggers expression of the dormant resistant mechanisms. However, it's now believed that a seemingly minor spontaneous mutation in the genetic code responsible for ERM expression may also be able to convert the inducible phenotype to the constitutive one in the absence of macrolide exposure. Thus, the bacteria is now also clindamycin resistant. Why this is particularly problematic is that conventional susceptibility testing will report the inducible strains as clindamycin sensitive. In other words, the inducible phenotype is technically clindamycin sensitive, but it has an unusually strong tendency to develop spontaneous clindamycin resistance. So is there a way that we can test for inducible resistance? Luckily there is. It's called the D test. Here's how it works. Take a plate of agar and add some of the Staph aureus bacteria one is interested in. Then place 2 antibiotic discs on it, similar to those from the disc diffusion method. One disc is the macrolide erythromycin, and the other is clindamycin. After a day of incubation, one of 3 outcomes can be observed. In the first, there is a ring of bacterial growth inhibition around both discs. This is consistent with the null phenotype. That is, the bacteria express no resistance to either erythromycin or clindamycin at any time. The next possible outcome is no growth inhibition around either. This is consistent with the constitutive phenotype, though could also be seen with combined erythromycin and clindamycin resistance due to mechanisms completely unrelated to MLSB resistance. Finally, in the last outcome, there is a circle of clearance around the clindamycin disc, but not the erythromycin disc, and the side of the clearance circle closest to the erythromycin is blunted, such that it looks like a letter D from above. This is evidence of the inducible phenotype. What's happened here is as the clindamycin has diffused outward into the agar and into the previously clindamycin-sensitive bacteria, growth is inhibited up until the geographic region where erythromycin has already reached. In the area where erythromycin reached the bacteria first, it triggers expression of the ERM gene, thus leading to MLSB resistance and an inability of clindamycin to inhibit growth in that region. Any staph strains displaying this result are said to be D-test positive. For D-test positive MRS strains, the CLSI recommends reporting them as resistant to clindamycin, as well as adding to the susceptibility report. This isolate is presumed to be resistant based on detection of inducible clindamycin resistance. Clindamycin may still be effective in some patients. You should note that it is certainly possible for the outcome of the D-test to have no inhibition around erythromycin and a ring of growth inhibition around clindamycin that is a perfect circle, lacking the D shape. In this circumstance, the resistance to erythromycin would be based on a mechanism unrelated to the ERM gene, and these strains would be considered non-inducible. The bottom line is that infections secondary to inducible MLSB community-acquired MRSA that are either severe, deep-seated, and or associated with high bacterial burden, such as sepsis, endocarditis, osteomyelitis, or an abscess, should not be treated with clindamycin. However, since the development of clindamycin resistance in these strains requires a spontaneous mutation, the chances of which are related to the degree of bacterial burden, uncomplicated cellulitis and other minor infections due to this bacteria can potentially be treated with Clinda. Infections due to community-acquired MRSA with a negative D-test can be safely treated with clindamycin if traditional susceptibility testing reports the strain is susceptible. Before I finally leave antibiotic susceptibility testing, I will quickly mention a few reasons testing might not be done by the microlab on an isolate. First, susceptibility of the isolated organism may be totally predictable from the bacterial species alone. Second, the isolated organism could be presumed to be part of the normal flora. Or last, the low number of colony-forming units might suggest it's either a contaminant or colonizer. Microlabs will usually still perform susceptibility testing in these circumstances if the clinician specifically requests it. For example, in the event a patient is severely immunocompromised, or in the event of a high suspicion that an isolated organism represents pathologic infection, despite it usually representing normal flora. I'll switch topics at this point and end the lecture by talking about the major factors that spread antibiotic resistance. They are, first, the transmission of resistant organisms between patients within hospitals. This may occur due to airborne organisms, contact with contaminated equipment or environment, or contact with the contaminated hands of healthcare providers. The second major factor is antibiotic overprescription. And the last factor is the use of antibiotics in animals raised commercially for food. The exact role of this factor remains quite unclear, but it's believed to be a non-negligible concern. Going back to that second factor, which is under the control of individual clinicians, how can antibiotics be overprescribed? They can be given for conditions that are unlikely bacterial in etiology. They can be given for conditions that are usually self-limited, as with non-invasive bacterial gastroenteritis. And broad-spectrum antibiotics can be given for infections that can be successfully treated with narrow-spectrum ones. There is definitely a link between antibiotic use and the emergence of resistance. Here is a graph of the occurrence of penicillin-resistant Streptococcus pneumoniae plotted against outpatient penicillin use in 17 different European countries. You can see that the Netherlands and Germany have the lowest occurrence of this resistant bacteria and the lowest use of penicillin, while Spain and Portugal have the highest rates of this bacteria and 2 of the 3 highest rates of penicillin use. Likewise, here's a graph for the pesky problem of quinolone-resistant E. coli. Iceland has nearly negligible risk of this resistance problem and the lowest rate of quinolone use, while Portugal has both the highest risk and the highest use. There are many factors that contribute to overprescription. Physician-related factors include diagnostic uncertainty, lack of knowledge, and an inability to closely follow up with patients to ensure that a conservative, antibiotic-free approach to patient illness has been successful. Patient-related factors include an inappropriate demand for antibiotics, which unfortunately does happen. Medication non-compliance, as resistance is more common to develop when the bacterial population has only been partially knocked down by a prematurely discontinued course of antibiotics. And self-medication, in which patients on their own used antibiotics for a non-bacterial illness. These antibiotics may be acquired from unused prescriptions from family or friends, may be leftovers from incomplete regimens as above, or can actually be acquired over the counter in many countries. Finally, the major problem is insufficient time to discuss the risks and benefits of antibiotics. I've found that most patients who initially request antibiotics for an inappropriate indication usually reconsider if I have the time to discuss the risks and benefits in detail. Unfortunately, when time is in high demand, it is often easier and quicker for the physician to simply prescribe the antibiotic than undertake this explanation. Physicians, particularly those in the US but possibly elsewhere as well, have a belief that when a patient requests an antibiotic, if one does not eventually prescribe it, the unsatisfied patient will just move on with their business to another doctor who will. However, I think this effect is probably overestimated by most physicians, particularly when time is taken to fully explain the reasoning of not prescribing an antibiotic or of prescribing a more narrow spectrum antibiotic than the one the patient requests. That concludes this lecture on antibiotic resistance. The next and final lecture will cover a variety of leftover topics, including determination of treatment duration and periprocedural prophylaxis.